The overall goal of this procedure is to analyze the development of the Purkinje cell dendritic tree under various experimental conditions. This is accomplished by first setting up organotypic slice cultures of the postnatal mouse cerebellum. Next, cultivate the slice cultures under various experimental conditions. For example, test the effect of mGlu1 agonists. Then, fix the cultures and reveal the morphology of individual Purkinje cell dendritic trees by anti-calbindin immunostaining. The final step is to analyze the Purkinje cell dendritic trees grown under control or experimental conditions. Ultimately, results from fluorescence microscopy of the immunostained Purkinje cells and the quantitative evaluation of the size and branching of their dendritic tree show that activation of the neurotransmitter receptor, in particular mGlu1, profoundly affects Purkinje cell dendritic tree development.
The main advantage of this technique is that almost the complete outgrowth of the Purkinje cell dendrites occurs during the culture period, and thus a very complex issue like development of a dendritic tree can be studied in an in vitro setting.
This method can help answer key questions in the developmental neurobiology field, such as how activity influences the growth and development of neuronal processes.
To begin this procedure, dissect the cerebellum from the brain of a P8 mouse pup in ice-cold preparation medium under a stereomicroscope. Next,